हेलो गाइस, आई एम शुभम आपको ट्यूटोरियल्स। आज हम टाइप्स ऑफ आरएनए एंड राइबोसोम्स देखने वाले हैं इस वीडियो में। तो इसके पहले हमने बेसिक्स ऑफ ट्रांसलेशन देखा। ओके, सो अभी टाइप्स ऑफ आरएनए एंड राइबोसोम्स वी विल सी। These are the ingredients, okay, important ingredients for the protein synthesis. So before we start, subscribe to the channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. So first of all, types of RNA. As we saw in the basics, three important types. Okay, means three types are there of RNA: mRNA, rRNA, and tRNA. Okay, it is very important. See, the order has to be remembered. We know ATM. Okay, ATM machine. So replace the A with R. RTM. Okay, rRNA, tRNA, and mRNA. So here R is the highest. rRNA is the highest one, and mRNA is the lowest one. So this is the order: R, T, M. Okay. So mRNA ट्रांसक्रिप्शन से mRNA मिलता है. Okay, right? Most of the mRNA have the sequence जिससे प्रोटीन्स बनेंगे. We know this. So ट्रांसक्रिप्शन से ही बनते हैं सब RNAs. Okay, but we know mRNA is having start codon, stop codon. Okay, as well as mRNA में RBS होता है. Okay, RBS. RBS means what? Ribosome binding site. Okay, RBS means ribosome binding site. rRNA is the ribosomal RNA. Okay, mRNA is the messenger RNA. rRNA is the ribosomal RNA present in ribosomes. Okay, and tRNA is the transfer RNA. Okay, transfer RNA which will bring the amino acid by reading the codons present in mRNA. mRNA में codons है, right? We know this. Codons को recognize करेगा tRNA. Okay, and bring the amino acid. So one by one amino acid specific. tRNA is also very specific for amino acid. Okay. Abhi first of all we will see the tRNA structure. Okay, because tRNA is a very important thing, right? tRNA will bring the amino acid. So it is having a very important role in translation. Okay. So this is a tRNA structure, very important. Okay, very important, right? Clover leaf. ओके, ऐसे बोलते हैं स्ट्रक्चर को क्लोवर लीफ मॉडल, राइट? सी, दिस इज हैविंग एंटीकोडॉन लूप. ओके, एंटीकोडॉन लूप जहाँ पे एंटीकोडॉन है. ओके, दिस विल बी 3, राइट? 3 विच विल रिकॉग्नाइज द कोडॉन, राइट? यहाँ पे कॉम्प्लीमेंट्री बाइंडिंग होगी mRNA एंड tRNA के बीच में. ओके, दिस इज द नेक्स्ट लूप, वेरिएबल लूप, right? Now this is psi-U loop, psi-U. Okay, psi-U means what? Pseudo-uridine. So pseudo-uridine. Okay, मतलब uridine जैसा ही structure होगा. Okay, we can say some changes. Pseudo-uridine. And here D loop is there. D loop. D stands for dihydrouridine. Dihydrouridine. And the next is acceptor arm. 3' CCA sequence is present on the acceptor arm. Okay, this is very important, right? Okay, so we have to remember anticodon loop and this acceptor arm with this sequence. These two are very important factors for the protein synthesis to occur. Basically, anticodon loop की वजह से complementary binding, right? Correct recognition of the mRNA codon, okay? Because that codon will code for a specific amino acid, right? अभी specific amino acid को code कर रहा है, तो हमें proper amino acid चाहिए, right? So this sequence, or we can say acceptor arm with this 3' CCA is very important for the proper amino acid to be coming. Okay, accurate amino acid यहाँ पे आके bind होगा 3' end पर. Okay, after this A. Okay, we have to remember this CCA. C is at the 5', A is at the 3'. Okay, and here the amino acid will come and attach at the 3' A. Right? अभी ये जो 2D स्ट्रक्चर है एक्चुअली दिस इज अ 2D स्ट्रक्चर clover leaf model. Okay, now the 3D structure is the L shape. Okay, inverted L, we can say L shape. Here also we can see anticodon. Okay, here is the anticodon stem, anticodon loop, same structure. See here now acceptor arm. Okay, acceptor stem, and here is the 3' acceptor end. Okay, so this is called a 3' acceptor end, which is having a CCA, right? और एक चीज है यहाँ पे, we have to remember this also. And discriminator base. Discriminator base is also very important for accurate amino acid to be coming. Okay, मतलब accurate amino acid को लाना है. So this is also very important. Discriminator base with this sequence, right? 3' CCA present on the acceptor end. Okay, so these are the important points. अभी इस पिक्चर में हमें कन्वर्जन समझेगा बेसिकली हाउ द 2D structures and 3D structure is there. This is a simple loop formation and this is a coiled, little bit coiled, right? We can see, and this is a L shape intermediate. Okay, L shape intermediate, and this is a fully L form. Okay, this is inverted L actually, all right? So this is about RNA. The third type of RNA is the rRNA, ribosomal RNA, but that is present in the ribosome. So we have to know ribosomes first of all. Okay, so let's see now ribosomes. So here we are starting with the ribosomes, right? Again, ribosomes, there are types. Prokaryotic ribosome 70S, eukaryotic ribosome 80S. Ribosome have small subunit, large subunit. So this is a small subunit, large subunit. Okay, 70S है in prokaryote. Okay, small subunit 30S, large subunit 50S. Okay, then in this C again here is the 5S RNA, rRNA, then 23S rRNA, then proteins are also there. Okay, small subunit contains 16S rRNA, 21 proteins. Okay, so this S stands for sedimentation coefficient. Okay, sedimentation पर depend है. Sedimentation coefficient in Swedberg unit. Okay, I will write here S is equal to sedimentation coefficient. इसका यूनिट है Swedberg. Swedberg, right? This is a eukaryotic ribosome. Eukaryotic ribosome is a 80S having large subunit 60, small subunit 40. Okay, then again large subunit में 5.8s RNA, 5s rRNA, then 28s, and the 49 proteins. Small subunit 18s rRNA and 33 proteins. See, now how to remember this? Okay, very simple. I will give you one trick, and the rest important factor also we will discuss in one slide. Two types: prokaryotes and eukaryotes. Right? Now in prokaryotes, known as 70s. Simple 70s, eukaryote 80s. Okay, this we have to remember: 70s in prokaryotes, 80s in eukaryotes. In prokaryotes, large subunit 50s, 30s. Okay, this is large subunit. This is small subunit. Okay, got it? So ऐसे ही हम याद कर सकते हैं 50s and 30s in prokaryotes, and in the case of eukaryotes 60s and 40s. 60s, 40s. Okay, in prokaryotes 50s, 30s, in eukaryotes 60s, 40s. One more we can do in eukaryotes. 6 plus 4 is 10. 60, 40 is 100. 100 minus 20 is 80. Okay, 100 minus 20 is 80. Okay, here what value comes? 80 minus 10 is equal to 70. Okay, but here what we can do, either we can just revise 50-30, 50-30 in prokaryotes and 60-40 in eukaryotes, right? Okay, as well as in the case of eukaryotes, total is 100 minus 20 is 80. We get. So this is confirmed in eukaryotes. If this is 80, this is 70. Okay, either you can remember one side completely eukaryotes and compare it with prokaryotes. It will be easy, or just keep on reading 70s, 80s, 50s, 30s, 80s, 60s, 40s. Got it? This is about ribosomes. Okay, one more thing we have to remember for ribosomes. Let's go to this prokaryotic picture. Okay, prokaryotic picture. Small subunit. Okay, first small subunit will come, right? And the 16s rRNA is very important. Okay, 16s RNA, rRNA is a sequence which will bind to RBS, ribosome binding site. Okay, ribosome binding site के साथ 16s rRNA आएगा. Okay, and it will bind to it with this small subunit 30s, right? And the next thing is the 23s rRNA is having the activity known as peptidyl transferase activity. Peptidyl transferase activity present on the large subunit. We have to remember this. So these are the very important things. Large subunit में है peptidyl transferase activity specifically related to 23s rRNA, and small subunit, that is 30s, is having 16s rRNA which will bind with the ribosome binding site. In prokaryotes, it is having another name. It is ribosome binding site having two separate names or sequences in the prokaryotes and eukaryotes. We will discuss this in the specific chapters of prokaryotes and eukaryotes respectively. From the eukaryote counterpart, 28s rRNA is having the peptidyl transferase activity. Got it? So these are the important things to be remembered. The next is the binding sites. Okay. So this is the site APE. We have to remember the sequence like this. APE. Spelling is ape. Okay, I will show with the comma. APE, ape. But we have to invert this. APE. Okay. So these are the three binding sites for tRNA. Three binding sites for tRNA. तो जब ट्रांसलेशन का प्रोसेस चालू है तभी tRNA इस तीन साइट्स पर बाइंड होता है. Okay, these sites are having their own functions, or we can say special what we can say activities. So उस accordingly इसको नाम दिया है A, P and E. Okay, so that we will discuss in the next video of translation specifically. So that will be more easy. Now the next thing about ribosome is the ribosome cycle. See, this is a ribosome cycle. This also ribosome cycle. So in the first step, we can see here, this is a mRNA with start codon and stop codon. Okay, start is given green color, stop by red. Then here is the initiator tRNA. First initiator tRNA will come and small subunit will come. Right? Also there are factors involved. This is just a ribosome cycle. Then the next thing is the large subunit will come. After small initiator tRNA and this large subunit will come. Means first small come. We just saw. Then the ribosome will be going, okay, moving in the 5' to 3' direction, and the amino acid will come one by one, one by one. So here is the next thing. Amino acid tRNA. Got it? Amino acid tRNA. See, this is the initiator tRNA. This is the amino acylated tRNA, and now polypeptide is synthesized. Basically, process is going on, and we can see here now the translation is going to end. This is the stop codon here. Protein synthesis has occurred. The next is the ribosome released, as well as protein chain released. Okay, basically peptide chain we can say, right? Then again this will be recycled. This is the mRNA. mRNA will combine with small subunit. Then the initiator RNA will come and the cycle will repeat. Okay, so basically cycle starts from here. This is the mRNA. Then small subunit will come. Then initiator, large subunit. Then this is known as scanning process. Amino acid tRNA. Okay. Then this is the elongation. Here is the termination. So this is just an easy explanation we can say, but there are a lot of factors involved, actually proteins. Okay. Now here, this is basically in prokaryotes. Transcription and translation occurring simultaneously. We can see here, as the RNA polymerase is transcribing the RNA, a ribosome is coming and started joining the proteins. Okay, protein synthesis is also occurring simultaneously. Right, मतलब ट्रांसक्रिप्शन ट्रांसलेशन एक साथ हो रहा है. जैसे जैसे ट्रांसक्रिप्शन हो रहा है आरएनए पॉलीमरेज से, वैसे यहाँ पे राइबोसोम्स आके बाइंड होगा. ओके, एंड बेसिकली दिस इज वी नो दैट दिस इज पॉलीसिस्टोनिक के मतलब बहुत ORFs, मल्टीपल ORFs आर देयर. नाउ हियर इज द पॉलीलाइबोसोम, or we can say polysome. This is also seen in prokaryotes basically. So this is what? Polysome. Okay, or we can say polyribosomes, because we can see many ribosomes are attached on the single mRNA. Okay, so this is a polysome. Okay, so that's it for this video about RNA types and ribosomes. We will start with the translation from the next video, actual translation process. These were all the, what we can say, requirements. Okay, and intro part of the translation, what are the necessary things. But still here, there are more things to be learned when we will see the translation process in prokaryotes and eukaryotes. As we will go, specific things will be understood basically. Like in the three binding sites, as I showed here. So this is having a specific function. Okay, and according to that, these names will be easy to be remembered. Why this is called P? Why this is called E? Why this is called A? So more answers we will get in the coming videos. So thank you guys. If you find this video helpful, do let us know in the comment section and subscribe to the channel and stay tuned for the upcoming videos. Thank you.